This is a video related with the book Statistical Modeling with R that I wrote and was published by Oxford University Press in 2023. And today I'm going to talk about experimental designs, starting with the obvious point of defining what is an experiment, then outlining the main principles of experimental designs, to be followed by a contrast between experimental designs and surveys and observational studies. And then the final point and longer of this video is an explanation of the main types of experimental design used in the life sciences. So an experiment is the deliberate manipulation of a system, that is, deliberate manipulation of the input of a system with the aim to quantify an output so as to infer the action of causal processes. Causal processes in quotes because we are going to infer the proximate causes of the behavior of a system. So we perform experiments because of the inherent variability of the response variables due to the heterogeneity of sampling units, the vagaries of measurement, chance events, and so on. This is to imply that we perform an experiment because whenever we change the input, we not always observe the exact same output. That is, there is variability in the output, and we want to use this variability to infer the potential causes of the observed behavior of the study system. Formulating any experiment will require us to define, first of all, the response variable that adequately reflects the behavior of the system, of the study system. Then the explanatory variable or variables that are to be manipulated or are to be controlled so as to properly infer the action of the causes of the observed behavior. And finally, these definitions stem all from the scientific hypothesis and knowledge of the system, and statistics only starts once these definitions are made. By this I mean that neither a statistic will not help us to define what is the proper response variable that reflects the actual behavior, the aspect of the behavior of a system that we want to study. Statistics does not tell us which are the best explanatory variables or variables that need to be manipulated in order to infer the causal processes affecting the behavior of the system. And statistics does not tell us which are the variable or variables that need to be controlled so as to make a proper inference of the behavior of the system. Statistics only works after we have made these three essential definitions. While comparisons and experiments have been carried out for centuries, it was Fisher's landmark book, The Design of Experiments, published in 1935, the starting point of modern scientific experimentation. There are three fundamental techniques of every experiment as devised by Fisher. These are, first of all, the replication. The idea here is to have is the repetition of parallel measurements of experimental units under the same set of conditions. The goal here is to reliably estimate summary statistics such as its mean, and to quantify more precisely their variation, that is the standard deviation or standard error depending on the case. Second point is blocking. The idea here is to isolate part of the variation of the response variable that is identifiable with a confounding variable, and we use this technique to isolate and control the effect of this confounding variable so as to make a better inference of the relationship between the explanatory variables and the response variable. The third point is the randomization. Here the idea is to prevent or to minimize the effect of systematic biases induced by the experimenter on the selection of the sampling units. The idea is to randomly assign the experimental units, individuals, plots, or what it be, to the different levels of the experimental factors in a random way so that we equalize the effect of the background variables affecting the response variable that we are going to measure. So observational studies, what they do is that they use the opportunistic selection of contrasted conditions. These contrasted conditions are selected so as to resemble the levels of one or more experimental factors that we would ideally would like to manipulate. So the observational studies are used whenever it is unfeasible, whenever it is unpractical, too expensive, or even illegal or unethical to purposely apply levels of a factor to experimental units. There are examples in the book that extend these explanations. So there are these are not true experiments because these surveys and observational studies do not apply the three main techniques that we discussed in the previous slide. So the main issue here is that observational studies cannot randomly assign the sampling or experimental units to levels of the experimental factor or factors that we are manipulating, and therefore these observational or studies or surveys may be subject to unknown biases that hinder the interpretation of results. Nevertheless, these surveys and observational studies are designed and analyzed with the same methods and ideas as if they were true scientific experiments. So the main type of experimental designs used in the life sciences. This is, of course, a personal a personal view of which are the six main experimental designs used in the life sciences, and this selection does not result of any systematic review of the designs applied in these areas. So most textbooks on statistics and experimental designs exclusively focus on the general linear model. This was the subject of the chapters 4 to 6 in the book and videos 5 to 10 in this series of videos. Yet all the experimental designs that we are going to talk about today and that can be conceived are valid for any response variable. So all the designs that I'm going to discuss in the coming slides will consist on two experimental factors, A1, A and B, each of which has two levels. Therefore, we have levels A1 and A2 and B1 and B2, and the 2 by 2 combinations resulting in the four combinations of A1 B1, A1 B2, A2 B1, and A2 B2. We are going to see that in a drawing in the coming slide. So I'm going to explain the Frequentist and Bayesian statistical models that will be fitted, and I will use the idea of the generalized linear model, depicting the response variable as if arising from one of the distributions that make up the exponential family of this distribution. The exponential family of distributions is characterized by a mean and a dispersion parameter whose meaning changes from one distribution to another. We should remember or recall that the response variable, the normal distribution, is one special case of the exponential family, and therefore one special case of the generalized linear model. So categorical variables that are going to be used, categorical explanatory variables modeled as random or group effects, will be used to indicate the experimental designs. So I'm going to specifically indicate how the R model that is going to be fitted in for each of the experimental designs. So we are also going to see the layout of the experiment and the general overview of the input data table for each experimental design. So the first and simplest design is the factorial design. It involves the simultaneous manipulation of levels of each of the two factors, and these are applied to every single experimental unit. That is, every single experimental unit is going to receive a combination of levels of the two experimental factors A and B that we are going to be manipulating. So let's have a look on the design. So experimental factor A and B are shown in white squares or gray squares, and experiment for white square for A1 and gray squares for A2, and the experimental and the combination A1 and B1 is going to be a white square. A1 and B2 is going to be a white square with an X, meaning that the level B2 was applied to this experimental unit. the level, the combination A2B1 is going to be shown as a gray square, and the combination A2B2 is going to be shown as a gray square with an X. So this sampling unit, this experimental unit, received the combination A1 and B1. This experimental unit received the combination A1 and B2. This one here received the combination A2B1, and this one here received the combination A2B2. So this is the general layout of the experiment, and here I'm simply showing for the first row, the first four experimental units. The data table is going to consist of one column labeled as A, one column labeled as B, and the response variable. Here we are simply identifying that this sampling unit received the combination A1 and B1, and this is the measured value of the response variable. This corresponds to this experimental unit, and we proceed in a systematic manner so as to complete all these experiment, so as to complete the table that has the values of the response variable and the values of the levels of the experimental factors applied simultaneously to each of these experimental units. So if we are to evaluate the interactive effect of the factors, there must be replication. That is, we must have replicates at the scale of the experiments, for each of the four combinations of the levels of the experimental factors A and B. So the statistical models, we are going to use a GLM. This is the response variable, and this indicates that we are fitting the single level effects of factors A and B and the interactions of factor B. This is in the Frequentist framework. In the Bayesian framework, the general command is BRM of the package BRMS, and we simply the statistical model has the same framework, and here the abbreviation XXX stands for the probability distribution that we are using to model the response variable. That is, this could be a Gaussian distribution, a beta distribution, a binomial distribution, gamma, negative binomial, and whatnot, each of which will have an associated link function. These ideas were described in previous videos, and I trust that you either remember them or have a look in the corresponding videos. For the Bayesian models, we also need to define the priors for each of the parameters in the same manner that we have been discussing in previous videos in this series. The second design is the randomized block design. Here, a block is a set of experimental units sharing a common set of effective conditions that we know beforehand that would affect the response variable and whose influence, therefore, we would like to quantify and control. So here, in dash line, we have a given block, a set of common conditions that are common to these four sampling units, these four experimental units, to which we applied the combinations A1 and B1, A1 and B2, A1 and B1, and A1 and B2. So these are so we need to therefore make up a data table in which we have the experimental factors A and B, the levels of which we apply to each of the sampling units, the block to which each of the sampling unit belongs or is associated with. Block is a categorical explanatory variable. So, and here are the values of the measured values of the response variable. So our model now has three categorical explanatory variables, two denoted as A and B, and the other denoted as block. So defining a block requires, of course, having background knowledge of this set of conditions that we know beforehand that would affect the response variable, and therefore, based on this background knowledge, we would like to control this the effect of this blocking factor or nuisance variable. So there must be replicates of each combinations of the levels of the factors A and B at the scale of the experiment, not necessarily at the scale of each block. So the statistical models, here I use the package GLM-TMB because this allows fitting just about any of the probability distributions associated with the response variable that we have been talking about in this series of videos. So here we have the factors A and B in interaction. This would fit the effect of factor A and factor B under interaction. This would be modeled as a fixed effects, and here we have block, the variable model as a random effect, and this one dash block, one vertical bar block, means that block we are going to be estimating a model in which we would have the intercept, random intercepts, and here family XXX stands again as indicated in the previous slide for any of the probability distributions that we would use to model the response variable Y. For the Bayesian model, the command here that we have been using is BRM of the package BRMS that would allow us fitting the model using the Hamiltonian Monte Carlo algorithm. Here we have the population level effects, and here we have the block level, the group level effects as defined by the categorical variable block. The statistical model is exactly the same as in the Frequentist framework. Here we would have also the name of the probability distribution used to model the response variable, and we would, as I said before, need to define the priors for each of the model parameters of this experiment. The split plot design is a design used whenever there is one factor, let's say A, that is too hard or too costly to change, and thus is applied on a large spatial scale. For instance, here I'm going to define also blocks. This is optional. So here we have a given block in dashed lines, and we have a given level of A1, here denoted by the long continuous line, to which we applied the level A1, and these are the smaller experimental units in which we apply the other factor B that are applied to smaller units that are nested within the larger units within in which factor A was applied. I recall that the blocking part here is completely optional. So the statistical model is the following. In the Frequentist framework, again, these are the fixed effects. We want to absolutely estimate the single level effects and the interaction between the experimental factors A and B, and here we indicate the random effects, in which we have a nested structure of the random effects to depict the actual structuring of experimental units in within the levels of A. So here we have one vertical bar block, if we have a block, dash A, this is going to be explained in the coming slide, and family, whichever family we use to model the response variable. In the Frequentist framework, the statistical model is written in exactly the same way. It's just that we use the command BRM of the package BRMS to fit the model. Let's have a closer look of this strangely looking group or random effects in the coming slide. So the random or group effects, one vertical bar block dash A corresponds to one dash block. That is, this is going to characterize the variance or the standard deviation, if you prefer, that would denote the variation among the blocks. This is an optional part. Blocks is an optional part, and the other component reflects the variation among the levels of the factor A within each of the blocks. This is a sort of interaction between two variables modeled as random effects. In the split plot design, the factor A is going to be modeled as both a random effect, random or group level effects, and as a fixed effect or population level effect. So a split plot actually involves the following sequential implementation. First of all, we define the large blocks. This is optional. Then we define the larger experimental units to which we applied the hard to change or hard to modify level of factor A, say A1 and A2, and within each of these larger experimental units we apply in smaller experimental units the level of factor B corresponding to the X or the absence of X in the previous slide. So the precise, we always need a precise description of the implementation of the split plot design in order to understand and be able to analyze it in a proper way. I recall that there must be replicate of each combination of factor A and B at the scale of the experiment if we are going to estimate the interaction between these two experimental factors. The nested design, in contrast to the factorial design, a given factor A is nested. A factor A is nested in factor B. A factor B is nested in factor A only if certain levels of factor B are combined with each level of factor A. This is going to be shown simpler in the design. So here we see, for instance, within the level A1 of factor A, we have levels B1 and B2 of factor B. Within the levels A2 of factor A, we have levels B3 and B4. Notice that we do not have the levels B1 and B2 for the of the factor B for the other level of factor A2. We don't have the combination A2 B1 or A2 B2 for the level B. So again, we need to identify, we need to have the following table in which we indicate the level of the experimental factors A and B that were applied to each experimental unit to which we measure the value of the response variable. Because here in nested designs we don't have every combination of levels of factors A and B, we cannot possibly evaluate the interactions between the levels of these experimental factors. So the statistical models here are using for the Frequentist framework, using the command glm-tmb. We are going to have the additive effects of factor A and B, not the interaction, for the reason explained before. And here we have as fixed effects, and here as random effects we have one vertical bar A2A double dot B, meaning the specific interaction or combinations of levels A and B that were used for each experimental unit. And for family we use the probability distribution that was employed to model the response variable. For the Bayesian framework, the statistical model is exactly the same as before. It's just that we use the command BRM and the family corresponding associated with this package, with the package BRMS. Again, we need to define the priors for all the parameters in the Bayesian model. So the repeated measure designs, here we have a different situation. Each experimental unit receives a single combinations of factors A and B, and this experimental unit is repeatedly measured over time. We are going to measure the same experimental unit many times across during the experiment. So here now we have a given experimental unit, this one here, to which we apply the combinations A1 and B1, and we measure this experimental unit, say, 4 times in a row. So this is the identifier of the experimental unit. These are the values, the time periods in which we measure this experimental unit, to which we applied the levels A1 and B1, so we need to specify them, and here are the values of the response variable for this experimental unit in each instance of time. So the consecutive values measured for each experimental units are not independent. Of course, this means that they are not replicates, and we would need to model their eventual autocorrelation. There is a correlation over time, for instance, using an autoregressive process of the first order. There are several options for this. Please have a look in the book for further details. So the statistical models for the Frequentist framework, again the command glm-tmb for the Frequentist framework. Here are the fixed effects in which we have the single effects of factor A and B and their interaction. And for the random effects, we here are specifying random intercepts, random slopes for the temporal dimension of our experiment, vertical bar unit, experimental unit. This is the identifier of each experimental unit. Is a categorical variable. It's a categorical variable for which we are going to specify random intercepts and random slopes in this experimental design. For the Bayesian framework, the statistical model is exactly the same with the specifications that I indicated, and for both Frequentist and Bayesian frameworks, we should model the temporal autocorrelation of the set of experimental units. Here, this variable, categorical variable model as either random effects or group level effects, indicate a set of measurements that are bound to be similar to each other, since this set of, in this case, 4 measurements were obtained in the same experimental unit over time. The crossover design is different from the repeated measure design. Here, each experimental unit receives not a single combination of the levels of factors A and B, but it receives a sequence of levels of the factors A and B, and is measured repeatedly over time. So here we have a given experimental unit that is measured in 4 different periods of times, in which we applied a given sequence of the combinations of the levels of experimental factors A and B, and this is the values of the response variable. We repeat this as many times as needed as to complete the set of situations. Here, what we do, the allocation of the sequence of levels of factors A and B is randomized for each experimental unit. It means that for this first experimental unit, we applied the combination A1 and B1, and then A2 and B1, and then A1 and B2, and then A2 and B2. But for the next experimental unit, we applied another sequence. This sequence is randomly applied to different experimental units. The autocorrelation over time must be specified and modeled as part of experiments. There are additional issues arising in crossover designs that are described and discussed to some extent in the book. These are the washout and the carryover effects. Please have a look if you are ever going to apply this experimental design to your data. So the statistical models are very similar to the ones used in the repeated measure designs. Here are the fixed effects of factors A and B plus the effects of time, which could be entered as either numerical variable or numerical or categorical explanatory variable. Here we are simply indicating random slopes for the combinations of factors A and B to each of the experimental units. And for the Bayesian framework, the statistical model is again the same. This is using the command brm instead of the command glm mtmb. I just wanted to point out that this the analysis and the interpretation of the crossover design is a little bit more delicate than the ones and specific than the other experimental designs, and you should handle this with a lot of care. So 3 final points before concluding this video. First of all, this is a very quick and shortened description of the main, according to me, experimental design using the life and environmental sciences. By no means this is meant to say that these are the only experimental designs that exist. Second of all is that these experimental designs that were here explained using just 2 experimental factors. They could be extended as to have more than 2 experimental factors, and they could include as 1 or more numerical explanatory variables as needed in each case to control the effect of these confounding variables. The final point is that the model of autocorrelations in the repeated measures and the crossover design may require some more complex model for certain data types, so please handle this with care and take a close look to my book or to any other book before analyzing this type of data. It is not difficult, it just needs to be done carefully. So in this video I have talked about experimental designs. I started by the definition of what is an experiment, the main principles of experimental design stemming from Fisher's 1935 book. Then a characterization of the surveys and observational studies in contrast to true scientific experiments, and ending up with the six main types of experimental designs used in the life and environmental sciences. I hope you found this video interesting. Thank you for your attention.